Good morning, everyone. Welcome back to my channel, Molecular Mysteries. Hope everyone is doing fine out there. So, without any further delay, let's get into the class.
Come and fly away with me. Come and fly away with me.
Good morning, everyone. Welcome back to my class. So, how are you doing? So, in the last class, we have already discussed about all the important metabolic pathways that happen in the cell, and the final important step that we have to conclude is the production of ATP, making use of all the NADH and FADH2 that are produced from the Krebs cycle and all. Okay, in today's class, let's discuss about oxidative phosphorylation. What is oxidative phosphorylation? So if you see with the sentence there, it's the oxidation of your carbon fuels and the phosphorylation for the conversion into ATP is what you call oxidative phosphorylation. So you have already gone through the glycolysis, Krebs cycle, beta oxidation pathways and all citric acid cycle previously, right? So you should be well aware of all the NADH produced, number of ATP produced, at what step FADH2 is produced and all in citric acid cycle, right? So the final step that helps in the production of ATP is the oxidative phosphorylation. So oxidative phosphorylation happens in the mitochondria in all eukaryotic cells, right? So you are well aware of the structure of mitochondria. In the cell biology class, I have also already covered that the mitochondria has got the inner membrane folded called cristae, and upon which you got the F0 F1 particles which are protruded out of your from your inner membranal mitochondrial membrane into the matrix. Thus F0 F1 complex is nothing but ATP synthase complex that I will cover in this class also. Okay, you should also be well aware of what's really happening and how the cellular respiration, how the aerobic process occurs in the mitochondria, how the oxygen is produced, and how the water molecules are produced in the oxidative phosphorylation. Okay, so before I enter into this, so I want to tell you this oxidative phosphorylation is also called as electron transport chain. Okay, the electron transport chain combined with chemiosmosis that happens in ATP synthase complex combines to form an oxidative phosphorylation. So this oxidative phosphorylation, also called the cellular respiration, I will tell you how this happens. Oxidative phosphorylation is a process in which ATP is formed as a result of the transfer of electrons from NADH or FADH2. NADH and FADH2 are the electron transfer potentials. They have high energy potential for the transfer of your electrons. So, the electron acceptors in the inner membranal mitochondria would accept them, making the protons electron motive force into a proton motive force. So that is how the gradient is created across the mitochondrial membrane. Let us take in a stepwise process. So don't get confused with what are the types of complexes in electron transport chain. Okay, now in this class I am talking about the functions of the electron transport chain and how each complex would convert the electrons and helps for the production of protons, forming a proton gradient. Okay, and in this class, in the next class, I will talk about the ATP yield and how many ATPs are produced for one molecule of glucose completely get oxidized. Okay, so in this class, let's all discuss about the detailed steps for the oxidative phosphorylation or the electron transport chain. Okay, so I told you this process takes place in the mitochondria in all the eukaryotes, as mitochondria is a major source of ATP in aerobic organisms, right? Oxidative phosphorylation compels the oxidation of carbon fuels to ATP synthesis. So the name itself says it's oxidation of carbon fuels for the phosphorylation of ADP into ATP. Okay, this oxidation and phosphorylation combinedly called oxidative phosphorylation that happens in the inner mitochondrial membrane. Okay, in short possible, I will tell you how this process happens. The carbon fuels are first oxidized into citric acid cycle for production of high yielding electron transfer molecules called NADH and FADH2. So you are well aware that in citric acid cycle there is a production of one molecule of FADH for one complete rotation of your TCA cycle, right? And also there is a production of NADH molecules for one round of Krebs cycle. So you will be getting the high transfer potential complexes like NADH and FADH2 which enter into electron transport chain. Okay. In oxidative phosphorylation, this electron motive force is converted into proton motive force and then into phosphoryl transfer potential. So there are four different type of complexes involved. These complexes are meant for the transfer of protons from the matrix side into the intermembranal space, so that those proton gradient would help you for the conversion of your ADP into ATP. Okay, let's take it in a stepwise process without any confusion. Okay, so students out there, please be aware that you should be well aware of all the complexes and where does NADH go for giving up those electrons and where does FADH go for giving up their electrons for the energy release. Okay, I will give you a detailed diagram. I will draw the diagram of the whole process, how the cycle happens in mitochondria. Okay, so I told you the conversion of EMF into PMF, electron motive force into proton motive force, is carried out by a respiratory chain consisting of three electron driven pumps, right? See, NADHQ oxidoreductase, Q cytochrome C oxidoreductase and cytochrome C oxidase. So you might be wondering, you told it were four complexes, why it is only three? Okay, you will be well aware of succinate dehydrogenase, an enzyme complex which is called meant for the conversion of your succinate into fumarate that you happen to see in the Krebs cycle, right? This electron transfer, this particular complex would take up the electrons but does not produce any protons into the mitochondrial space, intermitochondrial space. Okay, all these complexes, this is complex 1 and this is complex 3 and this is complex 4, all these complexes are meant for transferring of protons from your matrix into intermembranal space. But the succinate dehydrogenase enzyme complex, which is called as complex 2, will take the electrons but it does not produce any protons into a matrix, so it does not come under the electron motive force complex. Okay, so I will detailedly discuss you the process now. These transmembranes complex contains the multiple oxidation reduction centers like quinones, flavins, iron sulfur clusters and copper ions. So each and every complex has got these extra reduction centers. Okay, like iron sulfur reduction centers, copper ions and quinones, flavins, etc. Okay, so what are the four respiratory chains that I'm talking about in this class are complex 1, NADH oxidoreductase. So you are well aware that these oxidoreductase are generally, like you can even call it as NADH dehydrogenase. So removing of your hydrogen atoms, right? So these dehydrogenases enzymes would catalyze the oxidation reduction reactions which we have already discussed, right? So this is also called as NADH oxidoreductase. The second complex being succinate Q reductase, which is involved in the Krebs cycle conformation for the release of your FADH molecules, right? And complex 3 is Q cytochrome C oxidoreductase. I'm sorry, it was written QQ, it's only Q. I will it's my mistake, sorry. Okay, it's Q cytochrome C oxidoreductase and complex 4 is cytochrome C oxidase. Okay, so these are the four respiratory chains that contains of it. And today I want to before I give it in a theory part, I want to give you in a stepwise process so that you will be well aware of all the steps. Okay, so now let's draw the diagram. Okay, and this is your, okay, this is your intermembranal space and this is your inner mitochondrial membrane and this is your mitochondrial matrix. Okay, and now let's talk about the complexes I am talking about. This is your first complex, this is your second complex, this is your third, sorry, not good at drawing. And this is your fourth complex, and this comes the main important part, nothing but ATP pump, or you can say it as ATP synthase. Okay, right? Okay, this is your complex 1 I'm talking about. I'm writing here, it's your complex 2, this is your complex 3, and this is your complex 4. Okay, see, this is for your easy explanation and your easy understanding mechanism, how the cycle completely works out, how the cycle starts and end, and how this completely works. I am telling you here. Okay, and for your reference purpose, I will also give you the links in the description box for your further understanding. And the book that I am following for this process, or for the biochemical pathways or metabolic pathways, is Stryer Biochemistry. That's a wonderful book. Please go through it in the net or in the ebooks. Okay, so hope you got an idea. These are the 4 different complexes that I am talking about, and apart from these complexes, you have CoQ and cytochrome C here. Okay, so okay, now let us tell. I told you this is an NADH oxidoreductase, succinate oxidoreductase, and this is Q cytochrome C oxidase and cytochrome C oxidase. So you are well aware of the names. So now, from the glycolysis, from the Krebs cycle, from the beta oxidation pathway, you have all your NADH entering. So what happens is, this NADH enters the complex, give away its electrons to the first complex, gets converted into NAD+. Right? These electrons, once the complex receives the electrons, this complex gets excited, and the movement of protons takes place from mitochondrial matrix to intermembranous space. This is the first step that happens. So please remember that the NADH only enters into your complex 1 of electron transport chain. Okay, and this gradient is formed due to the more amount of protons present in intermitochondrial membrane, sorry, in its intermembranal space, than that of in matrix. Okay, and now once these protons are formed, the electrons now enter into give away those electrons to CoQ. Okay, these act as a cofactors for your movement of electrons from one complex to another complex. Okay, and once this CoQ gets the electrons get excited to CoQ. In the same way, after the transfer of electrons from first complex to CoQ, here comes the FADH part. This FADH would lose its electrons into the second complex, forming FAD+. These electrons, but upon taking the electrons, this complex 2 would never promote the proton, but it does not have the capability for converting the protons or movement of protons from matrix to intermembranal space. So there is no proton movement. Okay, so these electrons would immediately move into its CoQ. Okay, now this CoQ acts as an electron acceptor, where it accepts electrons from complex 1 and complex 2, and now it supplies to complex 3. These electrons, upon entry to the complex 3, get excited again like the complex 1, and it helps for the movement of the protons present in the matrix to your intermembranal space, and thus again there is a proton gradient formed across the membrane. So you have more and more of protons in intermembranal space than your matrix, and thus gives you a gradient. Okay, now this protons that have entered, and the electrons that are present in the 3 complex would move itself into cytochrome C. Upon entering into cytochrome C, this cytochrome C electrons in it get moved again into your complex 4. Upon the entry of the electrons into complex 4, this cytochrome C oxidase complex 4 would get excited like the 1 3 complexes. It would also help in the promote of your hydrogen ions from your matrix to your intermembranal space. So if you observe, you have tons and tons of protons accumulated in your in the intermitochondrial space than to your matrix. So this gives you a proton gradient. Okay, and this proton gradient, until here the electrons transfer has happened. Now the phosphorylation has to happen, and you know that ADP is a low energy particle, right? It has to be get converted or phosphorylated into a high energy particle called ATP, and for that to happen you need a lot of energy, right? To make it from a low to high energy molecule, you need to have an energy supply, and that high energy is supplied in the form of protons that are already there in your intermembranal space. And why this happens, why protons move, and you have already know that generally the protons move from a high generally the protons move from the high energy state to a low energy state to maintain an equilibrium, right? So for that it is coming down the hill from from high protons to a low protons. To maintain the equilibrium, protons need to be traveled through the ATP synthase complex, where it enters into the matrix again. So the proton gradient has helped the conversion of your ADP into ATP, because lots and lots of protons would produce a free energy. Okay, this free energy is required for the conversion of your ADP to ATP, which is meant for the production of your currency ATP. And this energy is produced in the cell. Like you can easily calculate how much amount of ATP can be released for oxidation of your glucose molecule. In the next class I will talk to you in detail at stepwise process how much amount of ATP is taken and how much NADH is produced, how many for a pair of electrons transferred from NADH, how much ATP, and for a pair of electrons traveled from FADH, how much ATP is produced. The yield will be discussed in the next class. Please go through the slides. Okay, so I told you the proton gradient would help you release of free energy, and this free energy is really required for the conversion of your ADP into ATP, thus producing happens to his electrons. These electrons that are present in the complex 4 would break down the oxygen into oxygen, and this particular protons that are already present in the matrix react with this, forming H2O. Okay, the oxygen molecule is broken down into two separate ions, and it is reacted with the already protons present in the matrix, forming the water molecule. So here oxygen is the electron acceptor. The electrons, all the electrons that are produced here are accepted by the oxygen, and it's converted into water molecules. So and you know what happens, this how the cycle continues. So you should be you are like there may be a doubt what happened to the protons that are produced here, and all the protons that are produced in the matrix again enter into the mitochondrial space, into the matrix, where the protons would be ready for the complexes like 1, 3 and 4 for the conversion into, for the movement into intermembranal space. Okay, so in short I want to complete this way. So let me tell you. Okay, so for your convenience I am giving you a brief explanation in short. So I told you the NADH that is already there released from all the biochemical pathways loses its electrons and enter into your first complex where the proton gradient is formed. Okay, and these electrons would enter into the CoQ, and after that the FADH that is produced from the Krebs cycle would enter into your complex 2, where it loosens its electrons, and it gets converted into FAD plus, and there is no proton movement, as I already told you. Okay, and now the electrons from CoQ get enter into the complex 3, where the conversion happens again. These electrons would excite this complex 3, forming the proton gradient and taking the protons from your matrix to your intermembranous space. Okay, and again this electrons that have been taken is now really, given into cytochrome C, and this cytochrome C would enter into the complex 4, where again the gradient is formed because the proton is moving again from your matrix to your intermembranous space. Okay, and now the electrons that have been there already is meant for the breaking down of your oxygen molecules into two oxygens that combine with the already protons present in the matrix, forming a water molecule. So at the end of this cycle, the more and more amount of protons get accumulated in your intermembranous space, and this gives you free energy, right? This proton gradient forms a free energy, and that is required for the conversion of your ADP into ATP. So you know that ADP is a low energy molecule that has to be that need energy supply for the conversion into ATP. So that energy is supplied by the proton gradient, and all the protons, as they generally the ground rule being the protons generally move from high energy state to a low energy state. It moves towards your low energy concentration and gives away all the protons, and these protons would again go back for your movement to your complex 1, complex 3, and your complex 4. So this keeps on continuing until all the NADH, FADH2 and all the things are done. Okay, this is how the cycle works. Okay, so hope I am clear with the complexes. Okay, and in this class I also want to discuss about, apart from this pathway, I want to discuss even more clear things, that like any questions that are posed where they may ask you what are the inhibitors. Like there are some antibiotics, or there are some drugs which would really interrupt your movement of electrons across the membrane, and what are those antibiotics? So rotinone is an antibiotic, or generally it's a drug that inhibits the complex 1. Antimycin inhibits the complex 3. Cyanide Co inhibits the complex 4, and oligomycin inhibits the ATP production. So for your understanding, I have given you this slide because there are some exams which may particularly ask you which enzyme, which antibiotic or which drug mainly damages the activity of your complex 1, complex 3, or okay. This is just for your understanding. Okay. Okay, so hope you got an idea about what the whole process is all about. Okay, so hope I'm clear with this class. So in the next class I will talk about the yield, complete yield of ATP. Okay, so see you in the next class, and thank you. See you in the next class. Until then, stay positive, stay healthy, and be kind to everyone. Thank you. Bye bye.